Hello everyone, I'm Kiran Kumar, fourth year PhD candidate from Biodiversity Research Center, Academia Sinica, Taiwan. And today I'm here to present on my research topic that is the evolution, pathogenicity, and the biosynthetic potential of genus Nocardia. Pathogenic bacteria are not just threats, but are also sources of valuable secondary metabolites. To unravel metabolic compounds and their associated biosynthetic gene clusters, also known as BGCs, requires comprehensive genome mining and comparative studies using multi-omic and bioinformatic tools and databases. Nocardia is a genus which comprises over more than 1,000 species, is known for its pathogenic diversity and ability to produce more than 50 bioactive compounds, which has clinical relevance. Due to the rising challenges posed by this pathogenic bacteria, that includes antibiotic resistance and limited treatment options, it is essential to explore their biosynthetic potential. Through this study, we aim to uncover novel bioactive compounds from the Nocardia genomes, which could serve as effective treatment for these infections. We used a comprehensive and integrated genome mining approach to study the Nocardia genomes, which were downloaded from the NCBI database, focusing more on the BGCs and their secondary metabolic synthesis. We conducted a pangenome analysis, evolutionary studies of BGCs, network analysis to examine the genetic diversity and connectivity within these gene clusters. Our results reveal that most of the genomes deposited in NCBI database for Nocardia, they mostly belong to the pathogenic category, and more than 40% of the species belong to group 1 pathogenic category, where these species are responsible to cause infections but are not that much harmful to the human beings. Based on the pangenome analysis, we saw that we have an open pangenome structure of 279,845 gene clusters. We identified more than 800 core genes, and more than 75% of the genes identified were species specific genes. This result suggests that the genus Nocardia has open pangenome structure and genomic plasticity. The functional profiling of these genes identified from the pangenome suggests that the core genome are most dominated by the translation and the ribosomal biogenesis genes, whereas the cloud and the new genes genome are mostly responsible to produce the transcription and secondary metabolite genes. We conducted the evolutionary dynamics of these BGCs, and we identified more than 8,000 BGCs where the type 1 polypeptide synthase, terpenes, ectoins, and the non-ribosomal polypeptide synthase were prevalent in all the genomes. We identified antipeptide class 5 and the proteosin gene cluster, and these two BGCs were the first to be reported in in Nocardia genomes from our study. This marks Nocardia has one of the most extensive collection of BGCs genera within the phylum Actinobacteria. Evolutionary analysis of these gene clusters reveal species specific diversity, and this variation reflects gene turnover and adaptive evolution in the genus Nocardia. Since our study was the first to identify the proteosin BGC in Nocardia, we conducted an in silico analysis of this proteosin BGC, and it revealed that 15 matching genes with substantial sequence similarity from our Nocardia genomes, which suggests there is a horizontal gene transfer from the species of Streptomyces albus. Now, this analysis revealed potential homology and functional similarity. In future, to confirm is present, we'll do a thorough analysis of UPLC MS mass to confirm the proteosin gene cluster in Nocardia salmonacida. The network analysis, which was conducted using Bigscape, were used to identify the gene cluster families and singletons within the genus Nocardia. This revealed a high network among the BGCs, and NRPS, that is non-ribosomal polypeptides, and polypeptide synthase for the most diverse gene cluster families. In future, we would like to experimentally verify the biological activities of these identified compounds, mostly related to the anti-cancer and antibacterial properties. We also like to conduct a gene knockout studies to confirm the biosynthetic pathways of the secondary metabolites and to also assess their functional roles. In conclusion, our research highlights Nocardia's remarkable biosynthetic potential and its dynamic pan-genome structure. Now, these findings not only advance our understanding of the genus genomic diversity, but also pave the way for the discovering novel natural products with significant pharmaceutical applications. Thank you for your attention.